morning. Thank you very much for the opportunity to present our... now you can hear me? Yes. You took 5 minutes from my time. Okay. Okay. So basically, what I would try to do today is present to you some of our recent work we done in the context of musculoskeletal development. And the first picture basically showing you a very simple diagram of the musculoskeletal system with the skeleton, tendon, and muscle. And the reason why I put these lines around each and every tissue is just to emphasize the fact that for, I think, a lot of time now, people were extensively studies each of these tissue, bone, tendon, and muscle, and really made great progress in our understanding in different aspects. However, when you think about it, the system as a whole was, or studied the system as a whole, was a bit neglected. And what we think in the lab is that there must be both molecular and mechanical interaction among musculoskeletal tissues that, because I'm more as a skeletal person, as it was presented here already, we think that that's a key feature or key point in regulating skeletogenesis. And what I will try to do today is to give you some taste of some of our studies that makes that or emphasize that work. And I want to say that in the end, that's one part of what is interested us. However, it is important to remember that in the end, we also know very little on how this system is actually assembled. I mean, we don't know how tendons are inserted into bones, and we don't know how muscles are connected into tendons. And I think this is a crucial and very important question that we will need to answer in the next few years. And just to show you that, or just to demonstrate the significance of the interaction within the musculoskeletal tissues during embryogenesis, there is a syndrome called fetal eknesia deformation sequence where from different reasons, the embryos basically, its movement is restricted, and as a result, there is many defects. And what is important for this talk is the craniofacial, limb abnormalities, and also joint problems. So if we are talking about joint problems, a few years ago, we were interested in understanding the contribution of movement, embryonic movement to joint formation. And we collected a group of mutated mice that either didn't have muscle development or were paralyzed. And a quick histological observation revealed that basically they lost their joint. And as you can see here, oops, sorry. As you can see here, this is the elbow. And you can, this is in the wild type, you can see radius, ulna, and when you look at the mutants, and it doesn't matter for that sense whether they lack muscles or paralyzed, you can see that the joint is basically missing. And the same for the shoulder, and this is just a picture analyzing all the joints in this embryo, and except the knee and some joints in the limb, in the fingers, all the joints were lost. We did a lot of work trying to understand what might be the mechanism for that, and one clue came when we start checking expression of genes which resemble differentiation into joint formation or to cartilage formation. And as you can see in the control, during that process there is a mutual exclusive expression of joint markers like GDF5 and collagen type 2, which resemble cartilage. You can see that in the elbow, basically there is a clear segregation between the two markers, showing that this is two different separate programs. However, in the paralyzed or in the muscleless mice, basically you can see that the joint space is actually cells express collagen type 2 together with while they're expressing the GDF5, the marker for joint formation, suggesting that they are like in this kind of mixed situation, or whether they are not following the right process. And to really pinpoint or to really show that these cells, that the joint cells lost their normal fate, we basically used a lineage study taking advantage of a mouse which expressed the Cree under the GDF5, which is, as you can see here, expressing joints. And once we did that in the background of our paralyzed or muscleless mice, you can see that, again, these cells basically descendants of the joint-forming cells. So you can see these regions which is express markers for chondrocytes are actually the cells which are originally were joint cells. And we summarize that by basically describing that a situation where normally you have in these joint progenitors, you have beta-catenin expression, which is activity, which is very important both for differentiating into the different lineages that eventually will form joint and to the self-renewal of these cells. Now, what we've shown is that in the absence of mechanical stimuli from movement, basically beta-catenin activity was going down in these cells. As a consequence, they lost their proliferation and their ability to differentiate into joint cells and instead become chondrocytes, and as a result, we lost the joint. And I think that it's a very nice example for the importance of movement and mechanical stimuli in regulating different progenitor cells in the embryo. So moving to another aspect, and that about the involvement of muscle load in regulating the shape of bones during embryogenesis, it is important to make just a small point that basically, although we understand or we have snapshots of the different bones on the different stages, we still miss the process itself, how they basically the morphogenic process. So let me just explain what I'm saying. So assume that this is bone in a certain stage, and this is a day or two later. So we have these two stages, but the question is how the shift took place. And of course, it could be like that, right? And then you assume a certain process of morphogenesis. However, it could be also in that way or that way, and of course that changed the whole process. So for that, to overcome this problem, we designed a specific program that can recognize specific structures in the mineralization, in the mineralized bone of the embryo, and as a result, we can now recapitulate or recover the morphogenetic process. And that's the picture showing you how if you align bones at stage an embryonic day 16.5 into bone at the stage of P6. I mean, there are many things that we can say about it, but I don't have much time. I mean, I just want to talk now about the ability of bone to acquire different circumferential outline. And as you can see, this is analysis of this process. And what is immediately obvious that there is a preferential growth. Some regions almost don't grow at all, while other expanded dramatically. And you can see that it's verified in different days as well. So now the question is, how now when we understand how bones are acquiring their periosteal growth, we could ask, what's the role of muscle in this process? And what you can see here is the mid shaft of two bones, femur and humerus, and this is a control bone, and this is a paralyzed bone. And what you can see is that, as I showed you before, there is a very specific outline to the different bones at different stages, which is obviously not a circle. You can see that in the paralyzed one, you ended up with a very symmetric bones. Namely, the bones develop evenly to all directions. And just to emphasize that, we outline the different circumferential outline of the different bones together, and you can see that each one of them has a very unique stereotypic shape. However, when you compare the paralyzed circumferential circumference, you can see that there is no difference, namely that muscle load is very important in that sense. And what we want to do, basically what we suggest, is to change the model where you have just very simple blue line into something which represent now more the reality better, and that is that bones basically develop in different direction, and by that basically acquire different cross-section. We believe that this cross-section is resulted from the increasing load that developing in the embryo, and to optimize the ability of these bones to carry up this growing load. I will skip that one, and just to quickly touch on another aspect of bone morphology, and that is related to the different protrusions, which is called eminence, which serve as an attachment site for tendons to connect the musculature with the skeleton. And I will not get into all the details. I will just summarize the work that we did. And we were able to demonstrate that, these regions are basically regulated by tendons and muscles, combining both molecular and mechanical stimuli, where initially the tendons, where there is a transcription factor called scaraxis regulating the BMP4 expression in the tip of the tendon, and by that inducing ridge formation. And then you need another signal, which is a mechanical one, which coming from muscle, and that growth depends on how tight the muscle pull, because in the absence of muscle contraction, this ridge will just initiate it, however, it will not develop. So let's see, okay. So just as a future direction, we are also very interested in going into a cellular level. And when we talk about cellular level, I think that the zebrafish fits very well for that. So we basically checked what's the effect of paralysis on zebrafish development, and we did it in several ways, and in all of them it was very clear that we looked on bones of the skeleton of the cranioskeleton, and we could see that there is a very big effect, namely the bones are shorter and basically miss shape. Here you can see the reason why we did this work, because as you can see, you can have a very nice cellular level, and one thing that immediately came into our, that we observed immediately, was the fact that if you paralyze the zebrafish, the chondrocyte lose their normal stereotypical morphology. And that basically hinted us into an explanation to this deformation that we saw in the skeleton, because there is a very important process, both in zebrafish and in our growth plate, which regulated the process of elongation, and that's chondrocyte intercalation. And as you can see, there is a situation where you have cells that change their morphology and then basically slide on one on top of the other, and by that you get elongation, and that's what you can see here in the bone. So we looked in the zebrafish by just paralyze this fish for a very short period, and you can see that what the consequence of that is accumulation of abnormal or roundish cells, and a lack of stacking process, suggesting that indeed basically muscle load is important for intercalation. And we went back to the mouse, and by measuring growth plate from paralyzed and unparalyzed mice, we observed that also in the mouse, the growth plate are shorter. And when we observed their cellular morphology, we found differences. And to make a long story short, we were able to identify that the columns in the paralyzed mice are shorter. As you can see over here, there are more columns with a restricted number of cells. And in the contrary, when you look on long columns in the wild type, you can see that there is clearly there is much more cells. And this is just to summarize this topic and to suggest that we believe that muscle load is important both in mice and in zebrafish for the intercalation process. So I just want to finish I think, in a very nice story, and that is about the sesamoid bones. So sesamoid bones is another part of our musculoskeletal system, and you all, I'm sure, recognize know your patella as an example for a sesamoid, but that's not the only place where you can find the sesamoids. We have it also in the hand and in the limb, and it's also their numbers vary in different organisms. For instance, frogs has many more. And what is interesting about them that they basically function to reduce the friction forces of tendons that go over joints, and also to increase the moment of the muscle. Now, when you look in textbooks how sesamoids bone are formed and why it is so interesting to understand how they form, because basically when you look at the histology, it looks like these bones stuck in the middle of a tendon. And that basically lead to two main hypotheses, which you can find in any textbook. First, that it's a mechanical stimuli sensed by the tendon, and the second is that the sesamoid formed within the tendon. So we decided to follow the sequence of how these unique bones are forming. And indeed, when we look late during development, you can see this is the sesamoid, this is the femur, and you can see that as you go lower, as you go down in development, the cartilage becomes smaller, and indeed it's stuck within or embedded within the tendon, which fits with the model. However, once you go around day 14.5, you can see that the patella is still, obviously can be observed. However, you can see that there is no space between the femur and the patella. There is another tissue there which is doesn't stain with Alcian blue. And when you look even earlier, you can see just a cloud of cells with no clear boundaries between the femur and the patella, suggesting that maybe the patella is actually originated within the femur. So that's very interesting, because it suggests that maybe you have a way to form new bones from already formed bones. And to try and understand how that process can take place, we follow different markers like SOX9 and COL2, which resemble chondrogenic markers. And what you can see between day 13 and 14, you can see that there is some kind of downregulation of these genes between the femur and the patella. And that resembles for us something, and this is just pictures that I took from another publication of us, showing you that when you form a joint, you downregulate COL2 and many other genes, and you upregulate GDF5. So that suggests that maybe the patella is forming simply by utilizing the process of joint formation. And we basically were able to demonstrate that by, again, looking on GDF5 as a marker and many other markers for joint formation, showing you that, that initially the region where the patella is forming is GD5 is not expressed there. However, when the patella is just on the moment where it's going to be separated, GDF5 is coming up, suggesting that indeed there is joint formation. So that clearly suggests that you can utilize or form new bones just by initiating a process of joint formation. So why people believe that mechanical load is important for making the patella? So we went back to our models, either the paralyzed or the muscleless, and as you can see, indeed, compared to the control, there is no patella. However, if you look carefully, it's clearly that that it might be that the patella is over here, but it's just maintained fused to the femur. And if you remember the way I started my talk, I told you that we were able to demonstrate that joint formation, it depends on muscle load. So that might suggest that that the patella is missing just because the joint program was not initiated. And for that, we went back and analyzed the expression gene expression of our paralyzed or muscleless or control mice. And what you can see very clearly, unlike in the control where you see the GDF5 coming beneath the patella, in the muscleless or the paralyzed mice, GDF5 is not coming, namely joint is not forming. And that suggests that, unlike was previously suggested that tendons, that the patella is forming within the tendon and then basically separated between the tendon and the femur, what we suggest now is that actually the patella is part of the femur, and at a certain moment there is initiation of joint formation, which basically remove this part from the femur, form articulation, and by that forming the patella. So just to conclude, I try to give you the impression that many aspects in skeletogenesis is depending on musculoskeleton bones and on tendons. And as I said, I think that it is now a big there are two big challenges. The first one is to be able to finish or to identify the exact parts that are in skeletogenesis which depending on these interactions, and then of course to be able to come up with the molecular mechanisms that regulate them, and then in the end or together with that, I think that the biggest challenge will be to really understand how the musculoskeleton is being assembled. Because this is like a central system in our body, and I'm sure that if you look in all the books or papers, this question is still open question and poorly studied. And I just want to finish with my team, and as you can see, if you want to be a member of my team, you have to engage your musculoskeleton system. Basically, yeah, so that's the team, and that's it.
This paper is now open for discussion. Please identify yourself.
Gérard Carcenti, New York. I was wondering, you made the point that the absence in the muscleless mice, this is the absence of movement that cause the absence of joint. But have you excluded completely the possibility that this is the absence of the molecular of the gene that is mutated in muscleless, that in fact cause the defect?
Right. So basically, at that work, we utilize 4 different genetic backgrounds, and to rule out the possibility that so that's one possibility that most chances that 4 different genes which known not to work in a pathway hopefully will not end up with some molecular influence. But in order to make it even clearer, what we did is that we took the PAC3 Cree mice and we crossed it into the ROSA reporter, and we show that in the joint and in all other places that we checked, there is no cell lineage which related to that. So I can't rule it out 100%, but I think that it's good enough in a way. But it is still, I can't rule that out. And there is another possibility, of course, that the muscles themselves secrete factors, which I hope maybe we will hear more about in this meeting, that also have some influence. So it's obvious that we need to be careful about that in a way.
Harris, NIA. So I had a question about the muscleless, the difference in the shape of what you were seeing in the shaft. And I wondered if you can do a major leap and then suggest that if we looked at the shaft of the humans, that weaker humans would have rounder shafts than people who are stronger. So have you actually ever looked at that?
Yeah. No, I myself, I didn't look in humans. However, I think that it is basically, if you look on papers, you can see that people who have some, and I'm sure that people here understand more than me on that, people who lost their muscle tone will change their shaft. I mean, what was surprising to us is two facts. One is that it has such a big influence during embryogenesis because we didn't suspect that. And the other one was that normally people assume that muscle load accelerate bone growth. And we showed that in some areas it's actually needed to repress bone growth. You're welcome.
So in these muscleless mice, you've shown that there's a major effect on the morphology of the bones and joint formation. And speaking of muscle factors, what about the development of other organs and tissues? Did you see any effect on those?
Yeah. So, so we basically didn't analyze that. I mean, it's like it's quite a challenge just to start understanding in 3 different tissues over one, and that represents just my restriction and not. It's definitely something that need to be done.
All right. Thanks very much.